AS Level Biology 9700. Paper 3, Variant 5, May June 2024. Question number 1. And question number 1 is, beetroot is a root vegetable that contains a red pigment in its cells. When beetroot is put in ethanol, the red pigment is released from the beetroot tissue and the ethanol changes to a red color. You know, beetroot has lots of color in it, so it is released when we put it into the ethanol. So you will investigate the effect of different concentrations of ethanol on the release of red pigment from beetroot tissue. Now you can see a table 1.1 in which they will give you labeled B, E, and W. And B will be the 4 beetroot cylinders in distilled water, and E 50% will be ethanol, and W will be the distilled water. Now, in if any solution comes into contact with your skin, wash off immediately under cold water. You should wear suitable eye protections. You will need to make different concentrations of ethanol, and you will need to prepare 20 ml of each concentration using E and W. And E is the ethanol and W is the distilled water. So in table 1.2 shows how to prepare the two of the concentrations of ethanol you will use. Decide which other concentrations of ethanol you will use. So here compare table 1.2 for the other complete. You have to complete this table. So they have given us two concentrations. So percentage concentration of ethanol, which is 50% and 0%. And on the other hand, we have to make 20 cubic centimeter or 20 ml of the solution containing distilled water and ethanol. So the first concentration which they have given us is the 50%, in which they have used 20 ml of ethanol and 0 water is there. The ethanol is already 50%. It means already it contains 50% distilled water. And the second concentration, lowest concentration, which they have given us the 0%. And at 0%, the volume of ethanol is 0 and the volume of water is 20 ml. Only water is there, ethanol is not present. So we have to make 3 more or 4 more concentration in between these. So first of all, I would like to make 3 or 4. It's up to you, but 3 are compulsory. There should be the total 5. 2 they have given us, and the other 3 which we will make. To make other concentrations, you can in the first concentration you can see that only the ethanol is present and water is not present. For this purpose, you can use a formula to calculate, to find out the other concentrations. For this, you will use this formula as, for example, volume of E. You need to know how much concentration. As for example, if we want to make the concentration 40, we want to make concentration 30, we want to make concentration 20. So for this purpose, you will need this formula, which I am going to write over here. The volume of E, which you need to find out how much ethanol will be added and how much water will be added to make solution, 30% solution of ethanol. So for this purpose, the formula is equal to, volume of E is equal to desired concentration. Desired concentration over 50, because we have to make concentration. So, and multiplied by 20. Now you must know what is 20. 20 is the ml of water which we have to make, ml of solution which we needs. we have to make a concentration of quantity 20 ml or 20 cubic centimeters. So desired concentration here in this case is now volume of E is equal to desired concentration is 40 divided by 50 multiplied by 20. 20 is that quantity which we want. Now after calculating this, it is 800 divided by 50. So it will be equal to, when we calculate it, it is equal to 16. So it means the E in this case will be 16. So we have to make total 20. So on the other hand, the volume of W which will be used will be 4 ml. 16 lm of ethanol and 4 ml of water. Same formula will be used to calculate the other concentrations as for example E, in which the E should be 30% and the volume, already the ethanol is 50% and 20% we have to make the volume. So it will be, so after calculating it is equal to 12. So here ethanol will be 12 and volume of water will be 8 because we have to make total 20. Again 20%, same formula will be used. So you can see that it will be 20 over 50 multiplied by 20. So it will be equal to 8. So volume 8, and on this side you will use the volume of water 12. So this is the very easy formula with the help of which you can calculate the concentrations or the volume of, to make a concentration of ethanol, the volume of the ethanol and the volume of the water which will be used to make these concentrations. Hopefully you have understood this, how to make these calculations. So now I'm moving towards the next. We have made these calculations. Keep in mind that we have to make total 20. So if we add all this 16 ml of water into 4 ml of water, so it will be total 20 ml of the solution. Again 12 and 8 will be again 20 and it will be again 20. So in this way, we are able to make different concentrations of ethanol by using distilled water. Now we will start our steps. Carry out step 1 to step 14. So carefully read all these steps when you are in the examination room where you will perform experiment. Uh, you have to carefully read these steps. First step is, in the beakers provided, prepare the concentration of ethanol as shown in table 1.2. So we have used this. That is, we have made 50%, 40%, 30%, 20%, and 0%. So label large test tubes with the concentrations of ethanol stated in table 1.2. So we will take large test tubes. 2, 3, 4, 5. So the very first will be 50% ethanol, 40% ethanol, 30% ethanol, 20% ethanol, and 0% ethanol. So now it is step 1, 2. Now step 3 is put 10 cubic centimeter of each concentration of ethanol into the appropriately labeled large test tubes. So 10 ml of E, 10 ml of the each concentration, those concentrations which you had made. That is, you will in this test tube number 1, 2, 3, 4, and 5. In test tube number 1, you will add 10 ml of 50%, in the 2, 10 ml of 40%, and in 3, 10 ml ethanol solution. Into test tube 30%. In 20%, you will add 10 ml of 20%, and in 10 ml of 0% into test tube 5. So in this way, you will set 5 large test tubes. Now step 4, cut the beetroot cylinders into 2 mm thick discs using a single edged blade. You will need 10 discs for each concentration of ethanol. As for example, if you have the cylinder, you will cut with sharp. Each should be of 2 ml. So I have not calculated this, so it should be 2 ml. Each disc should be of 2 ml thick. So you will need 10 discs for each concentration. 10 into 5. Total, you need total 50. You will put 10 into each. So start with glass rods. Put the discs into small beaker and cover with distilled water. As for example, you will add these 50 into this beaker with water, and then you will stir it. Pour the surrounding liquid into the beaker labeled for waste. So you will waste this one. You will waste in which the discs were present. Blot the discs on a paper towel to remove excess water. You can take a paper towel, and here you will put these beetroot discs and you will blot them. So put 10 discs into each of the large test tubes. Start timing and leave for 10 minutes plus 10 discs plus 10 discs plus 10 discs plus 10 discs plus 10 discs. So you will add 10 discs in each of these 5 concentrations. And you will note down the time, and you will keep it for about 10 minutes. For about 10 minutes. You will keep it for 10 minutes. Label small test tubes with the ethanol concentrations shown in table 1. Now you will make use of 5. 1, 2, 3, 4, and 5. 1, 2, 3, 4, 5. 5 more. So label small test tubes with the ethanol concentration. After 10 minutes, at the end, stir the contents of each large test tube. Pour the liquid from each large test tube into the appropriately labeled small test tubes. So you will add the liquid which was present in these large test tubes. You will add liquid from large test tubes. Only liquid. You will add up only the liquid. After 10 minutes, stir the contents of each large test tube. Pour the liquid from each labeled small test tube. The disc should remain in the large test tube. That is, you will add up only. Now you have to mark the color intensity by using this key. You can see over here. Yes. Shows the keys you need to use to record your results. If there is no color, 1 plus sign will be used. If very pale red color, 2 plus signs, 3 plus signs. As the color is increasing, the plus signs are also increasing. When there is no color, only 1 small plus sign is there. As the color is increasing, the sign plus signs are also increasing. So now you will observe the color of liquid in each test tube. You will observe the color in all these test tubes. And then what will you do? We are moving towards the next page. On the next page, you can see the next question is there. And next question is that you have to draw a table. Record your observations using the symbols. Record your observations in an appropriate table. So you will draw a table over here. So in this way, you will need to draw a table. On in one column, you will write the concentrations. You will write over here ethanol concentration. So it will be 50, 40, 30, 20, and 0, which we had used. Now, observation, observation, color intensity, observation, color intensity. So you have to write your answer according to your findings, according to the color, which you will observe in these test tubes in which you have poured the solution from the large test tubes without discs. After keeping the discs in them 10 minutes, for about 10 minutes. So maybe you will make use of these plus signs. You will make use of this plus signs according to your color intensity, which you will observe at that time when you are performing this experiment. So I'm not giving any plus or minus sign over here. So this is the finding which you will do when you will perform this experiment in the lab. So I will move forward. And when sometimes in some of my uh, papers, some of the students told me that you are copying the uh, marking scheme. There are some answers which should always be the same whether you are using marking scheme or not. Those are factual answers, and factual answers are always same wherever you are writing and from where you are getting those questions answers. So keep in mind, marking scheme is also compulsory for all this. Don't forget this. Marking scheme is also compulsory. So don't bother about this whether we have written the same answer or not. First of all, so describe the trend in your results. Again, what will you write over here? So this answer will be answer it after observing your findings or results, or the color you will observe, the color intensity which you have observed after your experiment. You will write over here the trend, so which trend has been shown over there. So with reference to your results, explain the effect of ethanol or membrane. Again, you will affect the you will answer according to your answer, or according to your results which you have done at that time. So little bit I would like to explain Otherwise, you have to write after observing your findings over there in the lab. So, explain the effect of ethanol on cell membranes. So, you would say that which you have observed during that experiment, then you will write all that, that what you have seen, what is the effect of that ethanol. You have used the different concentrations. So, after observing the results of those concentrations, that is, which concentration has shown the highest color intensity or which concentration has shown the lowest color intensity. So by using all these colors, you will explain. So what has affected the cell membrane? That is, which, the color is like, intensity is low and less color is there. So it means more cell membrane has allowed it to, the cell membrane acts as a boundary wall, which allow only some molecules to pass through it. All molecules cannot pass through it randomly. So in this way, so you can see that you, with the help of dot, you will explain this question. So the next question is state the dependent value variable in this investigation. Dependent, independent variable is that variable, as for example, uh, according to which the dependent variable change. So here independent variable is the concentration of the ethanol which we have used during this experiment, so different concentrations which we have used. And according to those concentrations, the dependent variable will always be the intensity of the color, which is the result, which has been changed. As the concentration has changed, the color intensity of the color will also be changed. So it will be varied at different concentrations of ethanol. So the dependent variable is the int intensity of color. Color intensity or intensity. Hope you have understood. State one variable that was standardized and describe it how it was standardized. Standardized means you have kept that, as for example, the here the different variables can be standardized. As for example, first of all, you have to use the you had to use the beetroots. So beetroots, the same size of the beetroots, there should be the same size of beetroots, there should be the same number of beetroots which you had used during this experiment. And at the temperature was also same. So here I would like to say that number of beetroots discs, size of beetroot discs, time for which you have kept, and another thing is the temperature, another variable. So these are the different variables, but we have to write only one. So preferably you can write this one. You can write others as well. They will be accepted. Now we are moving describe how you would modify. Let me Next question is how would you modify the procedure to investigate the effect of temperature on the permeability of beetroot cell membrane? Well, this is very you can say important, that how can you modify this? So modification is that we have want to know the effect of temperature on the permeability of beetroot cell membrane. It means we have to provide the different range of temperatures to the cell membrane, beetroot cell membrane, When we will keep this, when we will perform this experiment at different temperatures, so the color intensity will be different. It will be different with the same concentrations. So here we can set up the multiple temperature baths instead of just one temperature, as we were taking temperature 30 degrees centigrade in the previous experiment. Now here you can modify this by setup. We can modify by setting up a range of temperatures instead of using one temperature. You can use water baths, can be used for this purpose, electric water bath, to maintain different temperatures. And then you can perform the same experiment at different temperatures, and at the end of this you can see you can compare after when you will get the results. You can compare the modified procedure and modifying results of the modifying results and the other which you have already done. So it will give you a direct comparison of beetroot membrane permeability across a range of temperatures. So then you can compare the results of both experiments. So in this way, you can modify your experiments, and in this way, you will come to know that at which temperature the results they have shown the good results or the good color as well. Now, moving towards the next question, which is a graph which we have to draw. Researchers investigate the effect of drinking beetroot juice on blood pressure. Two groups of people were used in the investigation. One group was given 500 cubic centimeter of beetroot juice to drink. One group, where they drink. A control group was given water to drink. One group was given the beetroot and the other given the uh, the mean blood pressure of each group was measured at intervals. The difference in mean blood group pressure between the two groups was calculated. So shows the results of the investigation. So these results has been given over here. So on one thing we will take on x-axis and other will be taken on y-axis. So here in this case, the independent variable is the time and the dependent variable is, this is the independent variable. It will be taken on x-axis, and dependent variable taken on y-axis. So here you can see, except 0, all values are less than 0. That is minus 0.28. So the greatest value after 0 is minus 0.28, and the trend is in this range. That is you can see the smallest value is minus 0.92. And so in this way, I would like to say the and minus 1.33. So in this way, these values are very small values, which because they are less than 1. So we will move towards the next page, in which we will draw this. First of all, so we will take all these values on this. Let me do this a little larger, the page in this. So here I am going to draw the, so we will take the time on this side, time, and on this side we will take the difference in mean blood pressure. So the first value is 0 at 0. So first of all, we will make it, we will start right from here, 0, and then 50, and then it will be 100. One large square will be equal to 50 on x-axis. 150 and 200 axis. One large square which have 10 small squares will be equal to 50. And on y-axis, you know, one large square is equal to 0.2. So in this way, I am going to write the values over here. They are, we will start writing 0 at the top, because 0 is the largest value in this case. So 0 will be taken at the top, 0.0, and below this will be 0.28 or 0.2. You can take 0.2 and you can take 0.54, 0.4, 0.6, 0.8. Let me do it again, as my calculation are a little bit, we will start from the bottom. Little bit difficult for me to draw on screen of my laptop. So it will be very easy for you when you will draw in. So at 0, the value is 0. So we will, so always do this. Make a dot and circle it, or cross, make a cross. So here I am going to make the cross at 0, 0. So it will be here. Little bit difference can be observed in my drawing. And the next value is 0.28. So 0.28 will be equal to a little bit over here. 25. So it is on 25. So it will be in the center. It will be over here. And the next will be 0.5 at 80. So 80 can be over here. And then at 125 after this. So at 125, it will be about 0.92. So it will be greater than about over here. Sorry for this type of drawing, very difficult, but you can draw it very well, students, on a page. So at 160, so 160 will be over here, and 160 is 1.33. So it will be over here. And then again 0.87 at 220. So it will be little bit over here. So it will be minus, so it will be in this way. So now we have to draw it in this way with the help of scale. So you can see so a little bit double line is can be observed over here, but let me draw it again, or otherwise some students will think that there should be a double line. No, there should not be a double line, but you know, as I am drawing it without any scale, so it is little bit different with the actual. So this is the graph. Now in next question, you have to find out the value at 100. 100 you will go in this way. 100 you will be, it is near about in this way. So it will be 0.8 or something. So at 100. So this is the fine, because I will not come back now. I have already find this value according to this question. You can see after 100 minutes. So 100 minutes, the mean blood pressure for the control group was 15.79 kPa. So use your graph in figure 1.2 to calculate the mean blood pressure 100 times minutes for the group that was given beetroot juice. So here I have done this one. I have find out this value at 100. So it is about 0.9 or 0.8 something. So I'm going with it. Now I'm going to calculate with the formula, and that formula is, I'm going over there. So here in this case, the next page is, so we have to show over, so the difference, we have to find out the difference between these two values, which they have, that is the BP, BP for control, it is 15.79, APA, and on graph the BP for beetroot, beetroot group, it is about 0.87 from the graph. This is the value which we have got from graph. So the difference is equal to control BP minus beetroot BP. So control is 15.79 minus 0.87. So it is about 14.92. 14.92. So this is the answer. So it was all about question number 1. Watch it, watch the video, and also tell the other students about my channel, and like, share, and subscribe my channel. Thanks for watching. If you have any question, you can message me.